Remember, I told you we almost all organs of our bodies have a reservoir of stem cells. But these stem cells don't always come out and help us. So, how can we mobilize them? We have talked about signaling, cell signaling. So, is there any way we can signal these cells to come out and help repair the damaged organ? Let's look at an experiment which was done in my lab. This is actually a mouse model in which we caused myocardial infarction by ligating one of the coronary arteries. So the dead myocardium was placed in the tissue culture dish along with bone marrow mesenchymal stem cells. So here's the infarcted myocardium, this black dot, and around which you can see there's a cells, the bone marrow stem cells are swarming towards this tissue. So we tried different types of cytokines signaling molecules, IGF, GCSF, and actually we also tried SCF. So we wanted to see what combination will allow stem cells to move towards the damaged heart tissue. So when we use these different regimes, it turned out not only IGF and GCS help mobilize stem cells into the damaged tissue, heart tissue. Once these cells have reached the myocardium, they also start making proteins that are specific for the heart cells. So I would now like to show you controls of this experiment also. So here's the heart tissue, infarcted heart tissue. You can see that it is placed in a dish and you don't see the clumping of cells towards this tissue. Of course, you can see many cells here. It is because we allowed this experiment to go much further than our other experiment, which I showed you. So we didn't see any clumping, and these cells did not differentiate into heart cells, meaning that they weren't expressing specific heart proteins. So this is infarcted myocardium and mesenchymal bone marrow stem cells, but no cytokines. In this control, C2, we don't have the infarcted heart tissue, and however, we do have the cytokines. These cells, again in this control, did not form clumps and they did not express heart cell specific proteins. In C3, we have the chunk of the infarcted heart tissue. However, there are no cells, but cytokines were provided to this system. We didn't see any cells around it. We just wanted to ensure that there were no surviving cells which crawled out of the tissue and gave us the appearance of the clumping. In control C4, we have actually a heart tissue which was not damaged by myocardial infarction, and it was placed in the dish full of bone marrow stem cells in the presence of the cytokines we have talked about. These cells again did not form these clumps and they did not start differentiating into the heart cells. So what is the trick here? The trick, we think, is the ECM, extracellular matrix. Extracellular matrix of the dead tissue was also signaling these cells somehow to come to them in conjunction with the cytokines. And also once these cells reached there, with the help of ECM, extracellular matrix, these cells started producing heart-specific proteins, heart cell-specific proteins. To convince you, I'll show you another experiment. In this experiment, a muscle cell and nerve cell are used to demonstrate the importance of ECM. Here we have a system in which we have a muscle cell innervated by a nerve. We do this experiment two ways. We cut the nerve and we also get rid of the muscle. So in one scenario, we are going to allow the nerve to regenerate. Once the nerve regenerates, it goes and it finds the same, it synapses with the same region of the muscle cell which was making synapses. The nerve goes and forms synapses at the same spot where the original nerve was making synapses or contacting the muscle cell. So this is regeneration of just the nerve cell, the nerve and not the heart. So there must be something special about the ECM of the structure of the cell which is signaling this nerve to make contact here. Now if we don't allow the nerve to grow, we just allow the muscle to regrow, the muscle cells clump their acetylcholine receptors at the same spot where the nerve was originally innervating. Although there is no nerve there in this system, however, the acetylcholine receptors are clumping or concentrating at the exact same location where the nerve was previously innervating this muscle. So, meaning basically this implies that the extracellular matrix plays a very important role in signaling cells to do different things. And this is our hypothesis that ECM of the dead heart tissue was also signaling our bone marrow cells. So we think that the combination of signaling molecules and also signals being provided by the extracellular matrix helped cells express some of the proteins which are present specifically in heart cells, and the signaling molecules help the cells mobilize towards the damaged myocardial tissue.